Welcome to the session of NEET BYTE. So in NEET BYTE, what we are going to do, we will cover the topic from a NEET perspective. So we are just going to cover some very key points that you must remember for the forthcoming NEETs. So in today's sessions, we are just going to talk about Monera, right? Now, what is Monera? You know, Monera is the prokaryotic organization. Mind it, Monera can be unicellular or it can be multicellular. So, the very concept of multicellularity is not with the Monera. Take an example of Scion Bacteria. It can be unicellular, it can be multicellular. So, that's why the key thing what you have to keep in your mind is that Monerans are prokaryotes. Now, you see, basically, Monerans can be divided into two parts. What are they? They are Archaea Bacteria and Bacteria. Okay. Now, what are the bases on which this division has been done? There is only one basis, and that is the nature of the cell wall. In case of Archaea Bacteria, what happens? That cell wall is made up of polysaccharides. But in case of two bacteria or Bacteria, the cell wall is actually made up of the peptidoglycan. So that's why the entirely two different groups of the Monerans has been recognized. So let's first of all talk about the Archaea Bacteria. You know, Archaea Bacteria is an ancient bacteria. This is perhaps the first bacteria or the first living things that came into this earth, right? And they are also called as the living fossils. Now, why we call them living fossils? Because they haven't undergone any changes or very few changes in their structure since their evolution, since their inception to the life. So that's why we are calling them living fossils. Now again, the question is that why they haven't been undergone any changes. The reason lies in the very nature in the plasma membrane. Now what happens in Archaea Bacteria, the plasma membrane has a branch chain lipid, right? And this branch chain lipid doesn't allow them to interact with the outside environment. And you know very well, anything is going to change only when it is capable of interacting with the outside environment. Since it is not there, so that's why we do not see that the Archaea Bacteria has undergone any major changes. That is why we are calling them the living fossils. Okay, so two key things. One, the cell wall is made up of polysaccharides, and second, the plasma membrane has a branch chain lipid which make them environmentally very tough, and that is perhaps the reason why they are called as a living fossils. Now coming to the types of Archaea Bacteria. You know, the Archaea Bacteria can be divided into two groups. What are they? They are obligate anaerobe and facultative aerobe. Now what is obligate anaerobe? Obligate anaerobe are those groups which can survive only in the absence of oxygen. Now again, the obligate anaerobe can be divided into two parts, what we call methanogens and halophiles. Now what are methanogens? These are those bacteria which is capable of forming methane. They have a number of applications, like they are utilized in gobar gas plants or the anaerobic effluent treatment plants, right? In these two areas they are popularly being employed. As far as the halophiles are concerned, they are present in the marshy area, right? So they are also the obligate anaerobe. But there is another group, what we call them facultative aerobe. Okay, so basically they are anaerobe, but when oxygen is present, they start behaving as a aerobic respiration. Okay, and one of the best example is thermoacidophils. Now one of the nature of the thermoacidophils is that they survive at very high temperature, right, and at a very, very low pH. So these are the basic features for the Archaea Bacteria, right? This is the only things what you have to keep in mind as far as the Archaea Bacteria is concerned. Now quickly recap the important things in case of Bacteria or typical bacteria or true bacteria. Mind it, we call every bacteria as a true bacteria in which the cell wall is actually made up of peptidoglycan. So peptidoglycan is the criteria for telling any bacteria as a true bacteria or false bacteria. So when we study typical bacterial cells, first of all, we study that what are the structure or what are the protective structure that is present outside the bacteria. You can see in the diagram. So what happens that in a typical bacteria, the outer covering, or starting from outside to inside, we can say that outside there is a glycocalyx followed by cell wall followed by plasma membrane. Now what is glycocalyx? They are made up of polysaccharides. Again, glycocalyx is of two types. One what we call capsule, or one what we call slime layer, right? Capsule can be micro as well as the macro, and their size is 0.002 micrometer, right? And the slime layer is just a gel-like substances. The very objective of the slime layer is that it helps in attaching the bacteria to the host. That is the very objective, and also it's prevent the desiccation of the bacteria. So these are the two objectives for the slime layer. One of the best example of slime layer is the Paria disease. You might have heard about that. That in case of Paria disease, what happens? That bacteria stick to our gum with the help of slime layer, right? So these are the function of the slime layer. Now coming to the cell wall, perhaps the most important thing in the entire bacteria. Cell wall is made up of peptidoglycan. As the name suggests, two parts. One, peptide or amino acids. Second, glycan or amino sugar. So it is made up of two parts, amino acids and amino sugar. Now there are two types of amino sugar. What we call NAG or NAG, NAM or NAM. That is N-acetylglucosamine and N-acetylmuramic acid, right? And both are joined together by glycosidic bond, what we call beta-1,4 glycosidic bond. Okay. In addition to these two bondings, what happens that amino acids are present as a side chain, and there are 4 amino acids. What are they? That is L-lysine, that is typically found only in gram-positive bacteria. Okay. And in case of gram-negative, what happens? In place of L-lysine, there is going to have a diaminopymalic acid. So please remember these two names. It is often asked in NEET that which of the amino acids found in gram-positive and gram-negative bacteria, right? In case of gram-positive, what we see? The presence of L-lysine. In case of gram-negative, what we see? That there is a presence of diaminopymalic acid. So these are the only two names which at present I want to share with you. Now let's talk about that how we can differentiate between gram-positive and gram-negative bacteria. Now, what is gram staining? Now, gram staining is nothing but it is a crystal wallet, and there are 4 steps in which the gram staining is done. First of all, we put a stain, right? So once we put a stain, suppose we have taken two bacteria, one is gram-negative, one is gram-positive, and we are putting a stain in both of them, as you can see in the diagram. So what happens that the moment we put a crystal violet, both become violet. Then we put a iodine, right? And this is followed by the washing by alcohol. So what happens when we wash it by alcohol? One group of bacteria is going to retain the crystal violet and another group of bacteria is not going to retain the crystal violet. So the bacteria which is going to retain the crystal violet, we call them gram-positive, and the bacteria which fails to retain the crystal violet, we call them gram-negative. Okay, and then we do the counter stain by safranin. So when we do the counter staining by safranin, then the gram-negative bacteria is going to see the pink color. Okay, as you can see in the diagram as well. Now the question is that why there is going to have a differential staining, and the reason lies in the very structure of the gram-positive and gram-negative bacteria. Don't look at me, look at the diagram. So in case of diagram, you can see there that in case of gram-positive bacteria, what happens? The outside part is a peptidoglycan, and crystal violet is known for catching the peptidoglycan. But have a look at the gram-negative bacteria. In the gram-negative bacteria, what happens? Outside is a LPS, lipopolysaccharides. Okay, and there is a thin layer of peptidoglycan that is below the LPS. So the crystal violet fails to penetrate. Since it fails to penetrate, the color cannot be retained, right? So what is the big 3 difference between gram-positive and gram-negative bacteria? In case of gram-positive, the layer of peptidoglycan is more. In case of gram-negative, it is less. In case of gram positive, LPS is not there. In case of gram negative, LPS is there. In case of gram positive, periplasmic space is not there. In case of gram negative, periplasmic space is there. And gram positive forms exotoxin, whereas gram negative forms endotoxins. There is one more thing, and that is teichoic acid. Teichoic acid is actually present in gram positive, but it is not present in gram negative. So these are the few important differences what you have to keep in mind from NEET perspective. Now, from cell wall, let's come to the plasma membrane. Now, plasma membrane is a typical membrane, right? And the model for the plasma membrane was given by Singer and Nicholson, you know very well. So that is a quasi-fluid structure, which is a lipid bilayer in which proteins layers are actually embedded. So there are two types of protein layer, what we call extrinsic and intrinsic layer. But the important thing is that the plasma membrane of prokaryotes or monerans are quite different from the eukaryote on two aspects. One, in prokaryotic plasma membrane, what we see that cholesterol is absent, right? But in case of eukaryote, what we see, the cholesterol is present. That is the big difference. And second, the prokaryotic plasma membrane is going to act as a powerhouse of the cell, because we know there is no membrane bound organelle within the cell. So naturally they require ATP. So how they get the ATP? They get the ATP by the help of the plasma membrane only, right? So this is how we can differentiate between gram positive and this is how we can differentiate between the plasma membrane of prokaryote and eukaryote. Now coming inside the cell, inside the cell there is a general protoplasm, right? No membrane bound organelle, and we have a ribosome of 70S size, you know very well. 70S can be further divided into 30S and 50S, that you know very well. Now there is one more thing that is present within the bacteria, and that is some photosynthetic things, photosynthetic structures. You know, chloroplast is not there, but there are two groups of bacteria we can say. One, the cyanobacteria, and second, the photosynthetic bacteria, which are not cyanobacteria. So in case of photosynthetic bacteria, what happens, they have a structure what we call chromatophore. Okay, but in case of cyanobacteria, what happens? They have a thylakoidal like structure, right? So cyanobacteria are oxygenic, in which what happens, oxygen is released. But in case of non-cyanogenic bacteria, which do photosynthesis but they do not release oxygen, so they are anoxygenic. Okay, so all those structures in chromatophore is there, what we see that oxygen is not released. But in case of cyanobacteria, what happens? Oxygen is released, right? Finally, let's come to the genetic material of bacteria. As far as the genetic material, what we say, that genetic material is known as nucleoid or genophore. Now, how we can differentiate between the genetic material of bacteria and eukaryotes? One, circular chromosome. In eukaryote is a linear chromosome. Two, in bacteria histone proteins are not there. In eukaryotes histones are there. Three, in place of histone there is another protein what we call protamins, that is present in bacteria but not in eukaryotes. Four, the GC content, guanine and cytosine content in case of bacteria is quite high in comparison to the eukaryotes. So this is how we can differentiate between the prokaryotic and eukaryotic genetic material. And of course, needless to say that in case of prokaryote there is no nucleus, but in case of eukaryote, nucleus is always there. So this is how the general structure of the monerans we can discuss about. A typical eubacteria can be discussed, right? Very quickly, let's talk about the reproductions, how actually they reproduce. You know, the usual method of reproduction is the binary fission only, right? But the big question is there any sexual reproduction in case of bacteria? Yes and no. Answer is big no. Because you see, for sexual reproduction, the two criteria are needed. One, meiosis. Second, syngamy. Bacteria do not undergo for any meiotic division. So that's why we cannot say that bacteria undergoes the sexual reproduction. Of course, we use another word, what we call genetic recombination in bacteria. Right? So genetic recombination in bacteria occurs but not the sexual reproductions. Now there are four types of genetic recombination, what we call transformation, conjugation, transduction, sexduction. Now all these four names are nothing but they are just a mode of transfer of genetic material from donor to the recipient. So first of all, let's talk about how we can differentiate between a normal recombination and the genetic recombination in case of bacteria. For instance, if you talk about normal recombination, in case of normal recombination, what happens? That complete genome from male to female is transferred, right? This is what we call normal recombination, and as a result of transfer, a zygote is formed. But in case of bacterial recombination, please remember, complete genome is never transferred. Transfer of genome is always partial, or a part of genome is actually being transferred from donor to recipient. That is the moot point for the genetic recombination in bacteria. So that's why what we say that their zygote is not formed, but we use another word, what we call merozygote. So their merozygote is formed. Now the rest of the four names I told you, this is nothing but a mode of transmission. For instance, if I talk about transformation, what is transformation? Nothing. A transfer of naked DNA from donor to recipient. This is what we call transformation. Conjugation. What is conjugation? Nothing but transfer of genetic material from donor to recipient through conjugation tube. Right? When conjugation tube helps in the very transfer of genetic material from donor to recipient, we call it is a conjugation. And mind it, the conjugation tube is formed by the donor, not by the recipient. Right? And what is transduction? Transduction is nothing but it is the transfer of genetic material from donor to recipient through virus or bacteriophage. Okay, and finally, what is sexduction? Sexduction is the transfer through plasmid. Well, first of all, let me explain you the plasmid. What is plasmid? Plasmid is extra chromosomal genetic material. So, if I say extra chromosomal, what does it mean? It means that in addition to the usual nucleus or usual genetic material, there is another genetic material, what we call plasmid. So this is 1/10 of the genophore, right? Circular, and they replicate independently of the bacterial chromosome. Okay, and that is why they have a huge application in genetic engineering. Now coming to the sexduction, whenever the genetic material is being transferred with the help of plasmid through conjugation tube, then we call it is a sexduction, or we can say that sexduction is also a type or a part of a conjugation, because here conjugation tube is also involved, but the mode of the transfer is the plasmid. So plasmid actually take up some material of the host genome or donor genome, and they transfer it to the recipient. So this is how the recombination is going to happen. I hope the very concept of recombination is pretty clear to all of you, right? Now finally, let's talk about few of the monerans, and mind it, in these few of the monerans, only key point has to kept in your mind. For instance, take an example of Mycoplasma. What is Mycoplasma? First things, gram-negative bacteria. Cell wall is absent. Smallest size is 1.1 to 0.15 micrometer. They can cross the bacterial filtrate, popularly called as PPLO, pleuropneumonia-like organism. Okay, and they causes disease, what we call pleuropneumonia. Or in case of plants, little leaf of brinjal is another disease that is going to cause by Mycoplasma. This is the only thing what you have to keep in mind as far as the Mycoplasma is concerned. Now one more, actinomycetes, that is another very important monerans. Actinomycetes is a gram-positive bacteria. Mind it, question is always asked with reference to whether it is a gram-positive or gram-negative. So actinomycetes is a gram-positive bacteria, right? And it got a wider application in the preparation of antibiotic, or what we can say wide spectrum antibiotic. If I say wide spectrum, what does I mean? That the bacteria or antibiotics are prepared which can act simultaneously on the gram-negative as well as the gram-positive bacteria. That's why we are calling them a wide spectrum antibiotics. There is one more very important genera that is present in actinomycetes. right? And what is the genera? The name of that genera is Frankia. Frankia is known for the biological nitrogen fixation. So that also hails to the actinomycetes. One more important thing, actinomycetes is known as a connecting link between bacteria and fungi, right? Because they have some sort of mycelial-like structure. So commonly they are also known as ray fungi, right? This is the only things what you have to keep in mind. So just recap. Gram-positive bacteria, connecting link between bacteria and fungi, commonly called as a ray fungi. Wide spectrum antibiotics are prepared. Frankia, known for the nitrogen fixation. That's it. Now coming to another one, cyanobacteria. Now what is cyanobacteria? Again, first of all, it is a gram-negative bacteria, right? In case of cyanobacteria, the few terms or the structure that need to be kept in your mind. For instance, there is a one term what we call trichome, and another term what we call filament. Okay, another term what we call heterocyst. Three terms. Now what is trichome? Trichome is a filament structure, okay, in which there is a multicellular structure and they are arranged in a thread-like structure. This is what we call trichome. And if trichome is associated with a gel-like substances, then we call it as a filament. Okay. And if trichome is going to be fragmented, the fragments is actually called as a harmogonia. And harmogonia is capable of developing into new plant. Very important structure that is present in cyanobacteria is a heterocyst. Now what is heterocyst? It is a cell that is responsible for the biological nitrogen fixation. Why? Because there is a large quantity of nitrogenase enzyme is present over there. Okay, so this is all about the cyanobacteria, right? Cyanobacteria are commercially utilized for the preparation of biofertilizers, being because of their ability to fix the atmospheric nitrogen. This is the one thing, right? And they cause water bloom. Now what is water bloom? In any water, if there is going to have excessive growth of cyanobacteria, then we call it as a water bloom, right? So these are the things what you have to keep in mind as far as cyanobacteria is concerned. So let's recap the important point of cyanobacteria. First, gram-negative aerobic bacteria. Number two, it has a photosynthetic pigment in the form of chlorophyll A, right, and phycocyanin and phycoerythrin. Three terms: trichome, filament, heterocyst, harmogonia. Okay, no sexual reproduction. They always reproduce only by the fragmentations, right? Commercially, for the biofertilizers as well as for it is known for causing water bloom. So these are the only things what you have to keep in mind, right? One more, rickettsia. Now what is rickets? Again, it is a gram-negative bacteria, right? They are intracellular parasites, and we always say it is a connecting link between virus and bacteria, okay? And they cause Rocky Mountain fever, okay? So that is the only thing what you have to keep in mind. So once again, gram-negative bacteria, okay? They are naturally they are found in soil, so we can say they are heterotrophs in nature, okay? But when they infect, then they act as a intracellular parasites, okay? And we always call it as a connecting link between virus and bacteria. This is all what we have to discuss in case of Monera. So this was a neat quick bite. So whatever we have discussed in it, that is the minimum essential requirement to address the questions that is being asked from the Monera. See you in the next NEET bite. Thank you.